Hello, and welcome to today's session on LO1. Understand the features of prokaryotic and eukaryotic cells. Today, we will explore what distinguishes the simplest forms of life from more complex organisms. This session delves into the fascinating world of prokaryotic and eukaryotic cells, examining their unique structures, functions, and the essential roles they play in sustaining life. By understanding these fundamental differences, we gain insight into how life operates at its most basic level and how complexity arises in living organisms. Prokaryotic cells represent the most ancient and simple life forms. Found in organisms like bacteria, they are typically very small. These cells lack a true nucleus, with DNA freely floating in the cytoplasm. They do not have membrane-bound organelles. Despite their simplicity, prokaryotic cells are crucial to many biological processes. Prokaryotic cells have several defining features. They possess a rigid cell wall for structure and protection. Their ribosomes are smaller, aiding in protein synthesis. Plasmids carry additional genetic information. Some prokaryotes have flagella or pili for movement and attachment. Examples of prokaryotic cells include E. coli and Streptococcus pneumonia. These cells reproduce asexually through binary fission. This process is rapid, allowing quick adaptation. Prokaryotic cells are efficient and versatile. Their simple structure supports survival in diverse environments. Eukaryotic cells are more complex than prokaryotic cells. They form both unicellular and multicellular organisms. These cells are larger and contain a true nucleus. Membrane-bound organelles allow for specialized functions. Eukaryotic cells are essential for the development of diverse life forms. Eukaryotic cells have a true nucleus enclosed by a membrane. They contain organelles like mitochondria and the endoplasmic reticulum. The Golgi apparatus processes proteins, while lysosomes break down waste. A cytoskeleton provides structure and movement. Eukaryotic cells have larger ribosomes for protein synthesis. Prokaryotic and eukaryotic cells differ in size and complexity. Prokaryotic cells lack a true nucleus and membrane-bound organelles. Eukaryotic cells have a defined nucleus and numerous organelles. Both cell types share basic structures like ribosomes and cell membranes. These differences are crucial for their respective functions. Prokaryotic cells have a phospholipid bilayer cell membrane. Their cell wall, mainly peptidoglycan, provides shape and protection. The cytoplasm contains enzymes for metabolic reactions. The nucleoid region holds the cell's genetic material. Plasmids and ribosomes are essential for additional functions. Eukaryotic cells have a nucleus with linear chromosomes. The endoplasmic reticulum and Golgi apparatus manage protein and lipid synthesis. Mitochondria generate energy, while lysosomes digest waste. The cytoskeleton supports structure and transport. Chloroplasts in plants enable photosynthesis. Eukaryotic cells can specialize to perform specific roles. Cell differentiation allows for unique structures and functions. For example, red blood cells transport oxygen effectively. Nerve cells transmit electrical impulses rapidly. Each specialized cell structure supports its unique function. Red blood cells are biconcave, increasing surface area for oxygen diffusion. They lack a nucleus, maximizing space for hemoglobin. Their flexible membrane aids movement through capillaries, packed with hemoglobin. They efficiently transport oxygen. These structural adaptations optimize oxygen delivery. Nerve cells have long axons for impulse transmission. Dendrites receive signals from other neurons. The myelin sheath insulates the axon, speeding up transmission. Synaptic terminals release neurotransmitters for communication. These features ensure rapid signal transmission throughout the body. Muscle cells are elongated, supporting contraction and relaxation. Multiple nuclei facilitate high protein synthesis. Abundant mitochondria provide ATP for sustained contraction. Myofibrils contain actin and myosin for movement. These adaptations enable muscle cells to generate movement. Ciliated epithelial cells have hair-like projections called cilia. These beat rhythmically to move substances along surfaces. Goblet cells secrete mucus to trap particles. A thin surface allows efficient material exchange. These features protect vital organs like the lungs. Root hair cells have long projections for increased absorption. A thin cell wall shortens the diffusion distance. A large vacuole stores water and maintains osmotic balance. These cells lack chloroplasts. As they are underground, their structure supports efficient water and mineral uptake. Palisade mesophyll cells are packed with chloroplasts for photosynthesis. Their tall, rectangular shape allows close packing for sunlight absorption. Positioned near the leaf surface, they receive direct light. A thin cell wall facilitates gas diffusion. These features optimize energy production. The fluid mosaic model describes the plasma membrane structure. Proposed by Singer and Nicholson, it highlights a dynamic, flexible membrane. This model explains selective permeability and structural stability. The membrane's components move freely within a fluid-like layer. This arrangement supports various cellular functions. The membrane's basic structure is a phospholipid bilayer. Hydrophilic heads face outward, while hydrophobic tails face inward. This arrangement forms a stable barrier between cell environments. The bilayer's fluidity allows flexibility and self-healing. It acts as a semi-permeable barrier for molecule passage. Proteins are scattered throughout the membrane, like tiles in a mosaic. Intrinsic proteins span the bilayer, forming channels or carriers. Extrinsic proteins attach to the membrane surface. They play roles in transport, communication, and recognition. These proteins make the membrane selectively permeable. Cholesterol stabilizes the membrane by preventing fatty acid tail sticking. It maintains fluidity in varying temperatures. Carbohydrates attach to proteins or lipids, aiding recognition. They act as cell identity markers and assist in communication. These components enhance membrane stability and interaction. In conclusion, our exploration of the cellular world continues to reveal new insights and wonders. Thank you for your time and attention. Should you have any questions, simply send a text to 07484 300 and 61688, and a member of the UK Versity team will respond promptly. We value your participation and look forward to seeing you at the next session.